हेलो एवरीवन, वेलकम टू माय चैनल लाइफ साइंस ज्ञान। इस वीडियो में हम लाइफ साइंस सेट पेपर 2 के सॉल्यूशन देखेंगे, जो कि वेस्ट बंगाल पब्लिक सर्विस कमीशन का 2023 पेपर है। प्लीज़ लाइक एंड शेयर द वीडियो एंड सब्सक्राइब द चैनल। आइए शुरू करते हैं। हमारा पहला क्वेश्चन है, विच वन ऑफ द फॉलोइंग हैज़ बाई रेडियली सिमेट्रिकल बॉडी? प्लेनेरिया, फेसियोला, ब्रांचियोनस, मेट्रीडियम। तो हमारा सही आंसर है ऑप्शन डी। तो मेट्रीडियम, इट इज़ अ मेंबर ऑफ निडेरिया फैमिली फाइलम एंड हैज़ बाई रेडियली सिमेट्रिकल बॉडी। ऑप्शन डी इज करेक्ट आंसर। हमारा अगला क्वेश्चन है, मेम्ब्रेन फॉस्फोलिपिड्स फॉर्म बाई लेयर व्हेन वाटर on all sides due to amphipathic nature, presence of unsaturated fatty acid, presence of unsaturated or saturated fatty acid, asymmetric distribution of phospholipids, and also due to presence of cholesterol। हमारा सही आंसर है ऑप्शन ए, डी टू नेचर ऑफ amphipathic phospholipids form by layer when water is available on all sides. So there are two part of phospholipid, hydrophilic and hydrophobic tail. So it will form a bilayer where inside membrane hydrophobic tail will be there, and on outside you will see hydrophobic part. So it form bilayer by this nature। हमारा अगला क्वेश्चन है, विच वन ऑफ द फॉलोइंग इज नॉट स्टेप इन सदर्न ब्लॉट टेक्निक? सेपरेशन ऑफ डीएनए फ्रेगमेंट ऑन अ जेल, ligation of DNA into a vector, transfer of DNA fragment to a nitrocellulose membrane, hybridization of DNA with the labeled probe। तो हमारा सही आंसर है ऑप्शन बी। So in southern blot technique, you have to digest your DNA, then run a gel, transfer it onto a cellulose membrane, and you have to hybridize it with your labeled DNA, and by autoradiography you will see band of your interest। So this is known as southern technique, which does not involve ligation of DNA। So ligation of DNA involved in cloning process। So our answer is B, which is not a step in southern blot technique। हमारा अगला क्वेश्चन है, पॉपुलेशन डेंसिटी ऑफ अ इंसेक्ट increases from 50 to 60 in a month if birth rate for a period is 0.4, then death rate is, so these are the option। So our growth rate here is 20%, you can say 0.2। So growth rate is equal to birth rate minus death rate। So when you will calculate, you will see your death rate is 0.2। Our correct answer is option A। Our next question is, major goal of Golden Rice Project is to supply consumer in rice-based society with recommended daily intake of iron, vitamin C, vitamin A, vitamin D। हमारा सही आंसर है ऑप्शन सी। So golden rice are enriched with vitamin A। और अगला क्वेश्चन है, फॉर इंडक्शन ऑफ सोमेटिक एम्ब्रियोजेनेसिस इन हायर प्लांट द कल्चर्ड फ्राइबल कैलस टिश्यू नीड्स टू बी एक्सपोज टू हायर कंसंट्रेशन ऑफ ऑक्सीन फॉर शॉर्ट ड्यूरेशन, हाई कंसंट्रेशन ऑफ ABA फॉर शॉर्ट ड्यूरेशन, हाई कंसंट्रेशन ऑफ साइटोकाइनिन फॉर लॉन्ग ड्यूरेशन, लो कंसंट्रेशन ऑफ ऑक्सीन फॉर अ लॉन्ग ड्यूरेशन। तो हमारा सही आंसर है ऑप्शन ए, So callus needs to be exposed to high concentration of oxygen for short duration. हमारा अगला क्वेश्चन है ग्लूकोज 6 फॉस्फेट इज अ सबस्ट्रेट और अ प्रोडक्ट और इन ऑल ऑफ द फॉलोइंग पाथवेज एक्सेप्ट glycolysis, gluconeogenesis, beta oxidation, glycogenesis. Correct answer is option C. So except beta oxidation in all these processes, glucose 6 phosphate is either a substrate or a product. हमारा अगला क्वेश्चन है विच ऑफ द फॉलोइंग स्टेटमेंट इज करेक्ट रिगार्डिंग टेक्सोनोमी. सो दीज आर द स्टेटमेंट्स. हमारा सही आंसर है ऑप्शन डी. मीन्स ऑल स्टेटमेंट आर ट्रू. लाइक numerical taxonomy aims at determining phenetic relationship between organism. APG system of classification is established by taking the info from recent molecular studies. A phylogenetic classification is constructed in such a way that all the descendants of a common ancestor should be placed into same group, i.e. group should be monophyletic. So option D is correct. The next question is match the non-protein coding RNA molecules present in genome with their function. So these are non-protein coding RNA, means RNA does not form protein but they remains as RNA molecule and have function in different processes. So these are non-coding RNAs. So if you know function of some of the RNA, you can reach to your answer. Like cyst is a non-coding RNA which involved in X chromosome inactivation. So by this only you can reach to your answer. So for C, 1 is correct. So likewise, miRNA are involved in regulation of gene expression. No RNA is for mRNA splicing, and rRNA is involved in protein synthesis. No RNA are involved in pre-RNA splicing. हमारा अगला क्वेश्चन है विच वन ऑफ द फॉलोइंग कॉम्प्लेक्सेस ऑफ इलेक्ट्रॉन ट्रांसपोर्ट सिस्टम डज नॉट अकाउंट फॉर पंपिंग आउट ऑफ प्रोटॉन फ्रॉम माइटोकॉन्ड्रियल मैट्रिक्स. कॉम्प्लेक्स 1, 2, 3, 4. करेक्ट आंसर इज ऑप्शन बी. कॉम्प्लेक्स 2 does not account for pumping out of protons from mitochondrial matrix, means all others are involved. अगला क्वेश्चन है केमिकल that is required for maintaining protoplasts in intact condition during protoplast culture is DMSO, mannitol, PVP, sucrose. So protoplasts are nothing but plant cell or fungal cell without cell wall. So it needs to be maintained in a solution which will not rupture due to osmotic pressure. So mannitol is one of the osmolyte which maintains osmotic pressure in balanced condition. So correct answer is mannitol. This is used in protoplast culture to maintain integrity of protoplasts. The next question is individual with greater mass have a smaller surface area to volume ratio which helps in conserve heat. This rule is known as Allen's rule, Boltzmann's rule, Kopp's rule, or Gloger's rule. तो हमारा सही आंसर है ऑप्शन बी। Bergman's rule says that in colder region, if organism is having higher body, or you can say larger body, they will conserve heat. So like polar bear is having larger body, it helps in conserving heat. So this rule is known as Bergman rule. So option B is correct answer. हमारा अगला क्वेश्चन है, in a cell fusion experiment, a cell in S phase was fused with cells in G1 phase, G2 phase and M phase. The fused cells were grown in medium containing tritium labeled thymidine. So maximum radio labeled will be obtained in S phase cell fused with G1, G2, M phase, G2 and M phase. हमारा सही आंसर है ऑप्शन ए, बिकॉज इन G1 you will find DNA replication has occurred and cell is having 2n DNA, double DNA. So your S phase is already having double DNA, so this will fused with, if it is fused with G1, it will have maximum DNA labeled in this experiment. So A is correct answer. हमारा अगला क्वेश्चन है, in antibody molecule variable regions are present in heavy chain only, light chain only, both heavy and light chain, single heavy chain and single light chain. हमारा सही आंसर है ऑप्शन सी. If you see antibody structure, it is Y-shaped, having 2 heavy chain, 2 light chain. So on both a light chain and heavy chain, you will see variable regions are present. The next question is, which of the following is constitutive promoter for expression of transgene in a monocotyledon plant? 30S promoter, ubiquitin gene promoter, oleosin gene promoter, rubisco gene promoter. So according to West Bengal Public Service Commission, A and B both are right. So these promoter can be used for constitutive expression of transgene in monocotyledon plants. हमारा अगला क्वेश्चन है, मेजर डिटॉक्सिफिकेशन रिएक्शंस इन फेज 1 इन्वॉल्व्ड ऑल ऑफ द फॉलोइंग एक्सेप्ट ऑक्सीडेशन, एसिटाइलेशन, इपॉक्सीडेशन, हाइड्रोलाइसिस. हमारा सही आंसर है ऑप्शन बी. सो इट डज नॉट इन्वॉल्व इन डिटॉक्सिफिकेशन, अदर्स आर इन्वॉल्व इन डिटॉक्सिफिकेशन रिएक्शंस. Our next question is, which of the following diseases is associated with protein misfolding? So when there is protein misfolding, you will see accumulation of proteins. So options are cystic fibrosis, transmissible spongiform encephalopathy, phenylketonuria, Gaucher disease. So हमारा सही आंसर है option A, option B. So according to West Bengal PSC, option D is also correct, but in Gaucher disease there is non-functional glucocerebrosidase enzyme which will have higher accumulation of glucocerebrosides so it is not having protein accumulation, but you can remember this information for other exams. और अगला क्वेश्चन है, which one of the following statement is not true for retinal protein bacteriorhodopsin? It is major photosynthesis pigment of bacterium Rhodopseudomonas palustris. It converts energy of green light into electrochemical proton gradient. It functions as a light-driven proton pump transporting protons out of the cell. It is only protein of purple membrane Halobacterium salinarium. हमारा सही आंसर है option A. So this is not true regarding bacteriorhodopsin. It is saying that it is major photosynthetic pigment of bacterium Rhodopseudomonas palustris. So others are true regarding this protein. The next question is pygmy hog and hangul are the representatives of following states: Sikkim, Assam, Assam, Jammu Kashmir, Manipur, Sikkim, Mizoram, Tripura. सही आंसर है ऑप्शन बी. So pygmy hog is a very small pig which is representative species of Assam, and hangul, the kind of antelope, is a representative species of Jammu and Kashmir. Or you can say these are state animal of these states. हमारा अगला क्वेश्चन, विच ऑफ द फॉलोइंग स्टेटमेंट इज नॉट ट्रू अबाउट ब्राउन फैट टिश्यू? So it is abundant in mitochondria, it is specialized in heat generation, it is specialized for storage of fat, it is vital for survival of hibernating animals. हमारा सही आंसर है ऑप्शन सी. So this is not true about brown fat tissue. So brown fat tissues are abundant in newborn baby and also in animals which are hibernating. So in brown fat tissue there is heat generation which is having higher mitochondria and they are vital for survival of hibernating animals. हमारा सही आंसर है ऑप्शन सी. Next question is, in brackish water shrimp farm, a farmer detected some shrimps with white spots of variable sizes prominently seen on carapace. It could be caused by baculovirus, picornovirus, riovirus, parvovirus. हमारा सही आंसर है ऑप्शन ए. This is because of baculovirus. Our next question is, in a diploid organism, segregation of two alleles after a recombination event between the given gene and the centromere of chromosome occur at leptotin anaphase 1, diplotin anaphase 2. हमारा सही आंसर है ऑप्शन डी. This event occur at anaphase 2. हमारा अगला क्वेश्चन है, which one of the following serve as source of CO2, carbon dioxide for Calvin cycle in C4 plant? Oxaloacetic acid, malic acid, PEP, WBP. हमारा सही आंसर है ऑप्शन बी. So malic acid is a source of carbon dioxide for Calvin cycle in C4 plant. हमारा अगला क्वेश्चन है, which one of the following neurotransmitters is responsible for awakening from sleep? Serotonin, dopamine, acetylcholine or GABA? Our correct answer is option A, B and C. Here examiner have forgotten to add except, so except GABA, all these neurotransmitters are involved in awakening from sleep. Our next question is, acquisition of skill for playing piano is an example of non-declarative memory, declarative memory, sensory habituation, operant sensitization. Our correct answer is option A. So acquisition of skill for playing piano is an example of non-declarative memory. Our next question is methotrexate, an anti-cancer drug, is an inhibitor of DNA polymerase, dihydrofolate reductase, DNA topoisomerase, telomerase. हमारा सही आंसर है ऑप्शन बी. So methotrexate is anti-cancer drug, is an inhibitor of dihydrofolate reductase enzyme. The next question is which one of the following does not contribute in productivity in marine ecosystem? Phytoplankton, diatoms, dinoflagellates, algae, mollusca and crustacean. The correct answer is option D. So these are animals which are not involved in contributing in productivity in marine ecosystem. These are algae, or you can say kind of having chlorophylls, so they involved in productivity. Our next question, which one of the following statement about nucleolus is false? It is surrounded by lipid bilayer membrane. It appears due to synthetic activity of NOR region of chromosomes. It is site of synthesis of rRNAs. So rhizome assembly begins in the nucleolus. So in cell there is nucleus. In nucleus you will see a dark region, that is nucleolus. So this nucleolus is not surrounded by any membrane. So statement is not correct. So all are correct. So this is appears due to synthetic activity of NOR region of chromosome. It is site of synthesis of rRNAs, and ribosome assembly begins in the nucleolus. The next question, the aggregate mass of sterile cells located at inside base of capsule of Pelia is called eleuterophore, involucre, perigynum, pseudoeleter. Correct answer is option A. So aggregate mass of sterile cell located at inside base of capsule of Pelia is called eleuterophore. The next question, all of the following statements are true with respect to T lymphocytes except. So you have to identify false or incorrect statement. So T lymphocyte arrived from bone marrow and mature in thymus. During maturation, T cell acquire T cell receptor on membrane. The secretory product of T cell include antibody belonging to different classes. The cytotoxic T cells are also called T8 plus CD8 plus cells, as they have CD8 receptor on their membrane. So हमारा सही आंसर है ऑप्शन सी. So production of antibody is function of B cells. So all these statements are true for T lymphocytes except C. The correct answer is C. अगला question, if a Drosophila species has 3 pair of metacentric chromosome and 1 pair of telocentric chromosome, this species will have 7 arm in polytene chromosomes, 5 arm in polytene chromosome, 6 arm in polytene chromosome, 9 arms in polytene chromosome. So what metacentric chromosome have 2 arms and telocentric have only 1 arm. So if you will calculate 3 into 2 is 6 and 1 into 1, so total 7 arms will be there in polytene chromosome. So your option A is correct. Polytene chromosome is combination of all chromosome in Drosophila in a cell cycle. Our next question is, out of the following enzymes mentioned below, which one is activated by acid hydrolysis of proenzyme form? Pepsin, trypsin, chymotrypsin, elastase. Our correct answer is option A. So pepsin form from propepsin by hydrolysis? हमारा अगला क्वेश्चन है विच ऑफ द फॉलोइंग स्टेटमेंट इज नॉट करेक्ट फॉर फिनोमिना photoblastism. So photoblastism is a process of seed germination because of light exposure. So there are two light exposures, red light or blue light. So photoblastism. So phenomena of photoblastism was discovered in lettuce seeds. Germination of photoblastic lettuce seeds is regulated by pigment phytochrome. Phytochrome regulated lettuce seed germination is thought to be mediated by auxin. So red light irradiation induces the germination of lettuce seeds. तो हमारा सही आंसर है ऑप्शन सी, बिकॉज photoblastism involves function of gibberellin enzyme, phytohormone, not auxin. So option C is correct. All these statements are true for photoblastism except C. हमारा अगला क्वेश्चन है a phytohormone which is produced by plants under water deficient condition and plays a key role in tolerance response of plant to drought. Ethylene, abscisic acid, gibberellic acid, carnitine. हमारा सही आंसर है ऑप्शन बी. So abscisic acid, it is a stress phytohormone involved in tolerance response of plant to drought and other kind of stress such as salinity stress. So B is correct. Our next question is, which one of the following learning process belong to associative learning type? Habituation, sensitization, classical conditioning, postsynaptic facilitation. हमारा सही आंसर है ऑप्शन सी. So associative learning type is a type of classical conditioning. Our next question is, the restriction enzyme EcoR1 recognizes and cleaves at GAATC. What would be the approx number of DNA fragments produced by cleaving E. coli genome? So this is size of genome. So as you know, there are 4 nucleotides. So here the length of EcoR1 site is 6 base pair. So if you will calculate, you will find 4096 number of cleavage sites. So you have to divide genome size with this number 4096. You will find approximately 1123. 1123 is the answer. Option C is correct also. Next question is, if you wish to study the region of binding of transcription factor to a promoter DNA, which one of the following technique will be most appropriate? Chromosome walking, DNA footprinting, microarray, immunoprecipitation. Our correct answer is option B. So DNA footprinting is the technique to find binding of transcription factor to the promoter DNA. So chromosome walking is to identify promoter length, and microarray for relative expression of genes, and immunoprecipitation is to find protein-protein interaction, or you can say protein-DNA interaction. Our next question is, which one of the following statement about analysis of variance ANOVA is not true? So it is, ANOVA is commonly used for dividing the variance into components. It is used for determining whether the differences from mean are significant. The analysis of Analysis involved partitioning the variance and comparing the role of various factors, environmental, genetical, etc. It is calculated by adding the series of values and dividing the total by number of values. तो हमारा सही आंसर ऑप्शन डी. So ANOVA is a statistical method to know significance between differences from the mean, and it is commonly used for dividing the variance into components. The analysis involved partitioning the variance and comparing the role of various factors. So these are true statements, you can remember. The next question is, if a sample of DNA is found to have the base composition, mole ratios as follows. So adenine 40, thymine 22, guanine 21, cytosine 17. Then which one of the following conclusion will be most appropriate? So given DNA is single-stranded molecule, it is a double-stranded molecule, it has high melting point, it is retroviral genome. सही आंसर है ऑप्शन ए, because according to Kirchhoff rule, so your A, T, G, C. So if this rule is followed, your DNA is double strand. But in this case, if you add adenine and guanine, so you'll have 61, and if you add thymine 22 and cytosine 17, you will have 39. So you are not finding these are not equal, so your DNA is not double stranded, it is single stranded. So option A is correct. The next question, in red algae and in some Charophycean green algae, the elongated apical portion of female gamete that is receptive to male gamete is called trichogyne, trichome, trichocytes, tube cell. हमारा सही आंसर है ऑप्शन ए. So in red algae and in some Charophycean green algae, the trichogyne is a female gamete, portion of female gamete which is receptive to male gamete. This is trichogyne. Option A is correct answer. Next question, which one of the following statement is not correct? Hybridization between different species usually results in sterile offspring due to failure of pairing at meiosis. So cleistogamy is an outbreeding device. Sporophytic self-incompatibility results from genetic composition of tissues such as style and stigma, which prevents self-fertilization. Continuous selfing result reduced variation in a population and accumulation of deleterious alleles. तो हमारा सही आंसर है ऑप्शन बी. So cleistogamy, when your flower is closed and it has self-fertilization, so it is inbreeding device, not outbreeding. So your option B is not correct statement. B is correct answer. The next question is, what would be the frequency of occurrence of restriction site for PST1, which is 6 nucleotide site in a DNA from thermophile with GC content of 80% in its genome. So again, so frequency of occurrence of restriction site will be around 4096, and in these options you are having option A, which is near to this number, so option A is correct answer. Our next question is Agent Orange is used as a leaf defoliator by US Army during Vietnam War was 24D, Na, 24D और 245T, ethylene and kinetin. हमारा सही आंसर है ऑप्शन बी. These are synthetic form of ABA. So ABA is stress hormone, can cause defoliation in plants. This was used by US Army during Vietnam War and it is called Agent Orange. So option B is correct answer. The next question is which one of the following brain center in human is called troubleshooter in visual functioning? So lateral geniculate nucleus, amygdala, hippocampus, superior colliculus. हमारा सही आंसर है ऑप्शन डी. So superior colliculus in brain is known as troubleshooter in visual function. The next question is white-tailed deers are most active during twilight hours of dusk and dawn. Which one of the following circadian rhythms describe them? Diurnal, duskurnal, crepuscular, nocturnal. Our correct answer is option C. So this type of circadian rhythm is called crepuscular. Our next question, which one of the following cutaneous receptors is not activated by steady pressure stimulus? Cross bulbs, Merkel's disc, Ruffini's endings, Micinus corpuscle. हमारा सही आंसर है ऑप्शन डी. So these three are activated by steady pressure on skin, but this is not activated. Option D is correct answer. Our next question is following are the sum of statement given for dosage compensation. So you have to identify correct statements. So our answer is option D. 1, 2 and 5 statements are correct statements. Like in XX individuals, inactivation of one of the X chromosome occurs at early stage of development, not at one cell stage, and X chromosome inactivation is random. Any of two X, maternal or paternal, get inactivated, and females are mosaic for functional maternal or paternal X chromosomes. So these statements are true regarding dosage compensation. So you can remember these statements for other exams. The next question is in human usually predicted the half of babies born will be male and half will be female. Which one of the following is most appropriate explanation for this statement? So because of random segregation of X chromosome during female meiosis, because of random segregation of X and Y chromosome during meiosis, because all eggs contain X chromosome, because an average one half of all eggs produce females. सही आंसर है ऑप्शन बी. Because of random segregation of X and Y chromosome during meiosis, so you'll have half female and half male. So this process, the probability will be 50% for female and 50% for male. Next question, membrane potential in intact mitochondria will favor findings of which one of the following compounds after passage through the inner mitochondrial membrane? An ionophore, an electroneutral dye, cationic dye, anionic dye. हमारा सही आंसर है ऑप्शन सी. cationic dye. Our next question is, this is analytical question. So you have to identify or locate a gene on a chromosome. So there are different cell lines given, A, B, C, D, E, and the gene product is present or not present. There are two situations, and these are chromosomes present in cell line. Suppose in cell line A, you will see 1 to 7 chromosome are present, 8 or 9 is not. So if you analyze these situations, you will find that your gene is not present and that chromosome is not present. There are one condition. In case of chromosome 5, when it is not present, your gene activity is not present. In case of, for example, in chromosome number 7, when it is present, even then your gene product is not present. Means it cannot be on chromosome 7. Likewise, in chromosome 6, it is present but gene product is not present. In chromosome 4, it is present, your gene is not present. And in case of chromosome 3, it is not present, even then gene product is present. And in chromosome 2, it is not present but gene product is present. So in all conditions, the conclusion is the gene product, your gene is present on chromosome 5. So your answer is B, chromosome 5. So the gene is present on chromosome 5. So this is kind of mental ability question. हमारा अगला क्वेश्चन है, at which of the following sites partial pressure of CO2 is highest? Alveolar gas, systemic arterial blood, exhaled gas, systemic venous blood. हमारा सही आंसर है ऑप्शन सी, so at exhaled gas the partial pressure of CO2 is highest. Our next question is, which amino acid in a polypeptide undergoes N-linked glycosylation in lumen of rough endoplasmic reticulum? Serine, threonine, asparagine, glutamine. So for N-linked glycosylation, you will need extra amine group. So serine and threonine are having OH group, which are involved in O-linked glycosylation, and asparagine will have extra amine group, so this is involved in N-linked glycosylation. Your correct answer is option C. Our next question is, which one of the following marker systems reveal higher polymorphism? So smaller the unit, higher the polymorphism. So option A, RAPD, AFLP, ISSR, SNP. In all three, there will be involvement of different sizes DNA fragments, but here nucleotide variation is there. So this will SNP, a single nucleotide polymorphism, will have higher polymorphism. So option D is correct answer. Our next question, Drosophila melanogaster and Drosophila simulans are morphologically similar but reproductively isolated species. So they are known as sympatric species, allopatric species, sibling species, incipient species. हमारा सही आंसर है ऑप्शन सी. So these two Drosophila species are sibling species. हमारा अगला क्वेश्चन है ex situ bioremediation techniques involve excavation and subsequent transportation of pollutants to another site for treatment. Which of the following technique is not a part of this process? Bioreactor, biopile, windrow, bioventing. So if you are treating pollutants on same site where it is present, it is known as in situ technique. Or if you are taking out this pollutant and then removing pollutants, it will be called ex situ technique. So here a bioventing is kind of in situ bioremediation technique. So our answer is option D. All these processes are part of ex situ bioremediation. So where you will take out pollutants from a site and then treat on different site. Our next question is, there are two statements, one is assertion and another is reason or result. So assertion A says the carbon dioxide content of atmosphere has been rising in past few decades due to burning of fossil fuels. Reason or result is, on an average there is an increase in temperature of 0.4 to 0.5 degrees Celsius per decade. So both A and R correct. R is correct result of A. Both A and R are correct and R is not correct result of A. A is correct, R is incorrect. A is incorrect, R is correct. तो हमारा सही आंसर है ऑप्शन ए. So both A and R are correct and R is correct result of A. तो हमारा अगला क्वेश्चन है, which one of the following character of Anthoceros is shared with that of green algae? So stroma like slime pores, intercalary meristem at base of sporophyte, amphithecal origin of archisporium, each cell with single chloroplast having pyrenoids. हमारा सही आंसर है ऑप्शन डी. So this character is shared both in Anthoceros and green algae. In both, each cell with single chloroplast having pyrenoids. Option D is correct answer. हमारा अगला क्वेश्चन, इन E. coli the mismatch repair system repairs the misincorporated base which is not removed by the proofreading activity of DNA polymerase. While doing so, the repair system has to decide which DNA strand is newly synthesized, which DNA strand is parental. So misrepair system does it by recognizing GATC sequence nearby, specific repetitious sequences, hemi-acylated GATC sequence nearby, specific palindromic sequence nearby. तो हमारा सही आंसर है ऑप्शन सी. So DNA polymerase will recognize hemi-methylated GATC sequences nearby and will decide this is parental or newly formed DNA strand. So C is correct answer. हमारा अगला क्वेश्चन है, डिपोजिशन ऑफ पोलन ऑन स्टिग्मा of another flower of same plant is called? Homogamy, dichogamy, कैटोनोगेमी, जीनोगेमी। वहाँ सही आंसर है ऑप्शन सी, दिस इज नोन एज गैटोनोगेमी। Which one of the following option has characteristic of non-poisonous snake? Presence of cylindrical tail, broad transverse belly scales, fourth infraradial scale largest, and cylindrical tail, broad transverse scales, third supralabial touches eye and nose, presence of cylindrical tail and small belly scales, a cylindrical tail, broad transverse belly scales, and a loreal pit between eye and nostril. So हमारा सही आंसर है ऑप्शन सी, presence of cylindrical tail and small belly scales is a characteristic of non-poisonous snake. हमारा अगला क्वेश्चन है, a juvenile hormone JH regulates development and reproduction in arthropods. Chemically it is a derivative of amino acid, terpenoids, protein, flavonoids. हमारा सही आंसर है ऑप्शन बी, दिस इज अ टर्पेनॉइड। हमारा अगला क्वेश्चन है, म्यूटेबल collagenous tissue MCT is the characteristic of phylum Annelida, Arthropoda, Mollusca, Echinodermata. सही आंसर है ऑप्शन डी। So mutable collagenous tissue is characteristic of phylum Echinodermata. हमारा अगला क्वेश्चन, which one of the following technique would be the most suitable to identify microdeletion? G bending, R bending, fluorescence in situ hybridization, or inversion analysis? Our correct answer is option C. So which technique, fluorescence in situ hybridization, is most suitable to identify microdeletion in a DNA? Option C is correct answer. हमारा अगला क्वेश्चन है, fangs are specialized teeth used for venom deliverance in animals. In snakes, solenoglyphous fangs can be seen in vipers, kraits, cobra, hognose snake. Our correct answer is option A. So in vipers, solenoglyphous fangs can be seen. हमारा अगला क्वेश्चन है, baffles are metal strips attached radially to fermenter wall. The major function of baffles is to introduce sterile air in medium of the fermenter tank, break the vortex formed during the agitation process, introduce the anti-foaming agent in fermentation medium, filter the air that is coming inside the fermenter, and to prevent contamination. Our correct answer is option B. So baffles are metal strips. Their function is to break the vortex formed during the agitation process. हमारा अगला क्वेश्चन, which of the phytohormones is primarily involved in stomatal closure under drought condition? Again, so correct answer is option B, abscisic acid, which is a stress hormone. It is involved in closure of stomata. So your stomata will closed and will reduce water loss from plant and help in providing a drought tolerance to the plant. Option B is correct answer. Which moiety of hemoglobin molecule bind to 2,3-bisphosphoglycerate at pH 7? Amino terminal, carboxy terminal, sulfhydryl group, alcoholic group. हमारा सही आंसर है ऑप्शन ए। So amino terminal of hemoglobin molecule bind to 2,3-bis phosphoglycerate at pH 7. ऑप्शन ए इज करेक्ट आंसर। हमारा अगला क्वेश्चन है ग्राम नेगेटिव मोटाइल बैक्टीरियल स्पीसीज Pseudomonas carboxydoverans is an anaerobic carbon monoxide oxidizing bacteria, ammonia oxidizing bacteria, sulfur oxidizing bacteria, carbon dioxide reducing bacteria. Our correct answer is option A. So this bacteria is an aerobic carbon dioxide oxidizing bacteria. Option A is correct. So in an experiment with Xenophus blastula, some of the animal cap cells that are supposed to be ectodermal in fat are labeled with fluorescent dye and placed at the floor of blastocell. The labeled cells will be fated as endodermal, ectodermal, mesodermal, apoptotic. तो हमारा सही आंसर है ऑप्शन सी। So this will fated as mesodermal cells. और अगला क्वेश्चन है अ ब्लूप्रिंट फॉर एक्शन प्लान फॉर प्रोटेक्शन ऑफ द अर्थ एजेंडा 21 envisages scientific understanding of phenomena of climate change, biodiversity and conservation. Action plan for 21st century in all areas of environment and economic growth in a sustainable way. Global consensus on comprehensive management and conservation of forests. 21st meeting of Conference of Parties COP to United Nations Framework Convention on Climate Change COP 21. So हमारा सही आंसर है ऑप्शन बी। So Agenda 21 is an action plan for 21st century in all area of environment and economic growth in a sustainable way. So this is kind of environment science question. हमारा अगला क्वेश्चन है लैक्टिक एसिड कंपोनेंट ऑफ ह्यूमन स्वेट अट्रैक्ट्स मॉस्किटो एडिस एजिप्टी to have a blood meal. This chemical signal emitted by humans is an example of pheromone, caromone, cinnamon, elomone. हमारा सही आंसर है ऑप्शन बी। दिस इज a type of caromone. So pheromones are involved in sexual attraction. So like that you can remember. हमारा अगला क्वेश्चन है द फॉलोइंग स्टेटमेंट आर गिवन फॉर द जीन्स और जीन प्रोडक्ट दैट आर इन्वॉल्व इन गिविंग मॉलिकुलर एड्रेस टू द बॉडी पार्ट्स. So contains 60 amino acid homeodomain that fits mainly in the major group of the downstream genes, act as transcription factors, have 4 paralogous groups located in different chromosomes. The genes are clustered in 2 complexes. So which combination of above statement is best appropriate homeotic genes or gene products? So हमारा सही आंसर है ऑप्शन सी. So 1, 2 and 4 best appropriate for Drosophila homeotic genes or gene products. हमारा अगला क्वेश्चन है कटिंग ऑफ पोस्टीरियर रूट ऑफ स्पाइनल नर्व वुड impair motor control of skeletal muscle, interfere with flow of sensory impulse, interfere with the ability of brain to transmit impulses, interfere with circulation of CSF. तो हमारा सही आंसर है ऑप्शन पी। So cutting off posterior root of spinal nerve would interfere with the flow of sensory impulse. हमारा अगला क्वेश्चन है द फिश दैट लिव्स इन रिवराइन वाटर एंड बिकमिंग रिप्रोडक्टिवली मैच्योर माइग्रेट टू द सी टू ब्रीड देयर इज नोन एज यूरीहेलाइन फिश, स्टीनोहेलाइन फिश, एम्फीहेलाइन, पोटोमोटोकस फिश, एम्फीहेलाइन, थैलोसोटोकस फिश। हमारा सही आंसर है ऑप्शन डी। So the fish that lives in riverine water and becoming reproductively mature migrates to sea to breed. So this fish is known as amphihaline thalassotokos fish. हमारा अगला क्वेश्चन है टू लार्ज प्लाज्मिड ऑफ 80 kb and 100 kb have been isolated from rhizobium species. Which of the following technique should be most suitable to resolve these two plasmids? Phase, pulse gel electrophoresis, agarose gel electrophoresis, capillary gel electrophoresis. हमारा सही आंसर है ऑप्शन बी। For separating a large DNA molecules, pulse filled gel electrophoresis is best technique. Option B is correct answer. हमारा अगला क्वेश्चन है क्लेवेटा 3 CLV3 whoosh signaling pathway has evolved as the central regulatory pathway that coordinate stem cell proliferation with differentiation in shoot apical meristem, maintains adaxial identity in developing a leaf primordia, stimulates development of nitrogen fixing root nodules, alters the leaf size and shaping during development. सही आंसर है ऑप्शन ए। सो क्लेवेटा 3 एंड वुश सिग्नलिंग पाथवे कोऑर्डिनेट स्टेम सेल प्रोलिफरेशन विद डिफरेंशिएशन इन शूट एपिकल मेरिस्टेम। हमारा अगला क्वेश्चन है A 34-year-old man visits a physician with high fever, chill, severe headache, confusion. He has recently returned from Africa after completing an official assignment. A peripheral blood smear revealed multiple ring structures and crescent-shaped gametes. The most likely cause of complaint is Plasmodium falciparum, Plasmodium malariae, Plasmodium ovale, Plasmodium vivax. So हमारा सही आंसर है ऑप्शन ए। So this is because of Plasmodium falciparum infection. हमारा अगला क्वेश्चन है molten globule state is associated with nucleotide modification, lipid transport, carbohydrate synthesis, protein folding. हमारा सही आंसर है ऑप्शन डी। सो molten globule state is associated with protein folding. हमारा अगला क्वेश्चन है which one is a wrong match between the helminths and the disease transmitting invertebrate host? Guinea worm, snail, filarial worm, mosquito, liver fluke, snail, schistosoma and snail. So you have to identify wrong match. So option A is correct answer. For Guinea worm, you will see pig is host. All three options are correct. You can remember for other exams. और अगला क्वेश्चन है आफ्टर meiosis 1 each of the resultant daughter cells possesses twice as much DNA as gamete contains, four times as much DNA as gamete contains, half the amount of DNA as gametes contain. हमारा सही आंसर है ऑप्शन ए। आफ्टर meiosis 1 the resultant daughter cells will have twice as much DNA as gamete. So cell is in 2n state So it is twice than gamete DNA. और अगला क्वेश्चन है, इन स्नेल लिमनिया पेरेग्रा a male homozygous for dextral alleles S+S+ is crossed with female homozygous for sinistrial allele SS. So S plus is dominant and S is recessive. When F1 individual of this cross were crossed among themselves, means they are crossing S plus S with S plus S. So in F2 all progeny individual had a dextral phenotype, means ideally you should have this type of phenotypes, genotypes, and phenotype should be all around 75% should be with this phenotype, dextral phenotype. So the statements are, so this is, so in F2 all progeny individuals had dextral phenotype Y. So this is typical example of cytoplasmic inheritance, as the phenotype of female alone is being transmitted, exhibiting gene interaction with epistasis. The trait is quantitative and environment plays a crucial role to acquire the textual phenotype and exhibiting genetic maternal effect. तो हमारा सही आंसर है ऑप्शन डी. So in this case progenies are exhibiting genetic maternal effect, because dextral phenotype is phenotype gene is coming from mother, so it is affecting the phenotype. हमारा अगला क्वेश्चन है, which of the following pair of antibiotics interact with sterols in cell membrane to form channel through which small molecule leak from the inside of fungal cell to outside? Amphotericin and nystatin, amphotericin and bactericin, bacitracin and vancomycin, anisticin and cephalosporin. हमारा सही आंसर है ऑप्शन ए. So both amphotericin and nystatin forms channel-like structure from which small molecules leak from inside of cell wall to outside and fungus is killed. हमारा अगला क्वेश्चन है, if the overhead concentration of ozone at a point above the earth surface is 450 du, what is the thickness in millimeter of pure ozone at 1 atmosphere pressure? Our correct answer is option C. So that is 4.5. Our next question is, which class of chemical insecticides is characterized by a relatively high degree of environmental resistance? Carbamates, chlorinated hydrocarbons, synthetic pyrethroids, organophosphates. हमारा सही आंसर है ऑप्शन बी. So chlorofluorocarbon like components, or you can say fluorogens, which are having halogens, are more stable and having high degree of environmental resistance. So option B is correct answer. हमारा अगला क्वेश्चन है, a sample contains 4 proteins with following properties. So protein 1 is having molecular weight 14,000 and pi 10. Protein 2 is having 62,000 molecular weight and pi 4, and 3 is having 29,000 pi 8, and 4 is having molecular weight 9,000 pi 5. What will be order of emergence of these protein when mixture of 4 is chromatographed in anion exchange column at pH 7. So both molecular weight as well as pH is playing role in this process. So these are the options. Our correct answer is option B. So protein 1 and 3 will not bind to the column, and protein 4 will be eluted first, followed by protein 2. Okay. So protein 1 and 3 are having pH more than 7, so they will not bind to the column, and protein 4 will be eluted first because it is having less molecular weight than protein 2. So option B is correct answer. हमारा अगला क्वेश्चन है 3 different HFR donor strains are mixed with separate sample of F- strain, and the following mapping data are obtained from the studies of interrupted conjugation. So in this question, HFR donor strain is mixed with F- strain. So you have to map genes according to conjugation timing. So B gene is entering with 5 minutes. So this is supported by map in option A and option B. And option C, and your D plus D gene is entering in 10 minutes, means after 5 minutes. So your A and B is supporting this, and F is entering in next, C is entering in next 5 minutes, means total will be 15, and F is entering within 25 minutes. So your timing should match with entering of gene during conjugation. So here nearest answer or correct answer is option A. This is supporting this data. So this is type of analytic questions. You can understand and correct the answer. The next question is, when DNA is denatured by heating and subsequently allowed to reanneal, there will be an increase in absorbance at 590 nm, a decrease in absorbance at 260, an increase in absorbance at 260, and no change in absorbance and any wavelength. So हमारा सही आंसर है ऑप्शन बी. When DNA is denaturing, the absorbance will increase. When it is reannealing, your absorbance will decrease. So option B is correct answer. हमारा अगला क्वेश्चन है, P53 प्रोटीन इज एसोसिएटेड विद ऑल ऑफ द फॉलोइंग एक्सेप्ट ट्यूमर सप्रेशन, प्रोग्राम सेल डेथ, ट्रांसक्रिप्शन, post translational modification. Our correct answer is option D. So P53 is a tumor suppressor gene. It is involved in tumor suppression, PCD, transcription. So not in post translational modification. Option D is correct answer. और अगला क्वेश्चन है, which of the following statement is not true about telomerase enzyme? It is ribonucleoprotein. It prevents telomere shortening. It is active in all cells. It adds TTAZZ repeats to chromosome ends. हमारा सही आंसर है ऑप्शन सी. So telomerase enzyme, it is active in all cells. This is not true. और अगला क्वेश्चन है एनालिसिस ऑफ एक्सप्रेस्ड आरएनए इन अ सेल इज नोन एज प्रोटियोमिक्स, ट्रांसक्रिप्टोमिक्स, इंटरेक्टोमिक्स, मेटाबोलोमिक्स. Correct answer is option B. The analysis of expression of RNA in a cell is known as transcriptomics. So for protein it is proteomics. If you are studying protein-protein interaction it is interactomics. If you are analyzing metabolites it is metabolomics. Next question. Which of the following statement about Taq polymerase enzyme is not correct? It has 5' to 3' polymerase activity. It has 3' to 5' exonuclease activity. It is a thermostable enzyme. It has temperature optimum at 70 to 75 degree centigrade. सही आंसर है ऑप्शन बी. So Taq polymerase is not having 3' to 5' exonuclease activity. It is not true regarding Taq polymerase enzyme. All are a true statement. हमारा अगला क्वेश्चन है विच ऑफ द फॉलोइंग पेयर ऑफ एंटीबायोटिक्स इनहिबिट बैक्टीरियल सेल वॉल सिंथेसिस बट स्ट्रक्चरली डू नॉट कंटेन फोर मेम्ब्रेन membered beta lactam ring. Vancomycin, bacitracin, cephalosporin, ampicillin, phosphomycin, cephalosporin, ampicillin, methicillin. Our correct answer is option A. So these two antibiotics, vancomycin and bacitracin, they inhibits bacterial cell wall but not contain four member beta lactam ring. Others are containing beta lactam ring. So option A is correct answer. और अगला क्वेश्चन है विच ऑफ द फॉलोइंग स्पायरोचीट इज अ human pathogenic and designated as stealth pathogen. Spirocheta, Arontia, Trypanosoma pyramidum, Leptospira biflexa, Treponema succini faciens. और सही आंसर है ऑप्शन बी. So Treponema pallidum is human pathogen and designated as stealth pathogen. हमारा अगला क्वेश्चन है Histone acetylation increases transcription of gene because it increases DNA-histone interaction. Acetyl group on histone are recognized by DNA polymerase. Histone acetylation loosens the DNA-histone complex and making it more accessible to RNA polymerase. And histone acetylation increases DNA binding, which is recognized by RNA polymerase. So for histone acetylation हमारा सही आंसर है ऑप्शन सी. सो ड्यू टू एसिटाइलेशन हिस्टोन लूजन्स and the DNA histone complex will be loosed and it will be more accessible to RNA polymerase for transcription. Option C is correct answer. हमारा अगला क्वेश्चन है बैक्टीरियम दैट ऑक्सीडाइजेस रिड्यूस सल्फर कंपाउंड लाइक HS minus SO3 2 directly to sulfate with no intermediate is Acidithiobacillus ferrooxidans, Paracoccus, Pentotrophus, Pseudomonas, Strugeri, Brocadia, Amoxidans. A correct answer is option B. So bacteria Paracoccus pentotrophus is directly oxidizing these compound HS minus and SO3 2 directly to sulfate without any intermediate. Option B is correct answer. और अगला क्वेश्चन which of the following is accurate description regarding T cells positive and negative selection in thymus? It leads to deletion of T cells with high affinity for self-MHC peptide. It leads to deletion of T cells with low affinity for self-MHC peptide. It results in diverse T cell population with high affinity to self-MHC peptide. It results in diverse T cell population with low affinity to self-MHC peptide. The correct answer is option D. So T cells positive and negative selection in thymus results in diverse T cell population with low affinity to self-MHC peptide. D is correct answer. हमारा अगला क्वेश्चन है आइडेंटिफाई द एंजाइम दैट हैज 4 different variants each consisting of metal center containing any one of the following. So either copper, zinc, manganese, Fe or nickel. So catalase, glutathione reductase, glutathione S transferase, superoxide dismutase. The correct answer is option D. So superoxide dismutase containing two these metals. So this is correct answer. Which of the following statement is not correct for botulinum toxin? So it is one of the deadliest neurotoxin known to man. It is best produced in aerobic acidic environment with extremely high concentration of salt and sugar. Extremely minute quantity picograms are potentially lethal in adult human. So botulinum toxin blocks neuromuscular transmission by blocking the release of acetylcholine. So हमारा सही आंसर है ऑप्शन बी. So botulinum toxin is best produced in aerobic acidic extremely high. This is not true. So this is correct answer. All others are true statement regarding botulinum toxin. So you can remember these information. हमारा अगला क्वेश्चन है which one of the following is isoenzymes? Lactate dehydrogenase, citrate synthetase, hexokinase, aldolase. The correct answer is option A and C. So lactate dehydrogenase and hexokinase both are isoenzymes. So these two are correct. हमारा अगला एंड लास्ट क्वेश्चन है लाइम डिजीज is the most prevalent arthropod-borne disease in United States and is caused by infection with a Rickettsia rickettsii, Spirochete, Borrelia burgdorferi, Mycoplasma, Spiroplasma citri, Mycobacteria, Myxococcus flavus. The correct answer is option B. So Lyme disease is caused by Spirochete, Borrelia burgdorferi. So thank you for watching the video. Please like and share the video and subscribe the channel for more such video important for different exams such as SAT, CSIR NET, Assistant Professor exams. Thank you.